A key element of the UK Spine's mission is to support development of drugs which target biological drivers of aging. Toward this goal, the UK Spine's Bridge program has funded the project Identifying Factors that Restore DNA Repair in Aging Cells through WRN Reactivation. The principal investigator is Professor Lynne Cox of the Department of Biochemistry at the University of Oxford. Collaborating on this work is Dr. Ivan Bubriak, UK Spine postdoctoral scientist at the University of Oxford. Premature aging is seen in a number of human heritable syndromes that result from mutation in key DNA repair factors, strongly suggesting that failure to repair the genome is causative in driving aging and age-related disease. Evidence supporting this hypothesis comes from studies of normal aging, where senescent cells are found to accumulate with age and cause at least some of the diseases of aging. Senescent cells accumulate DNA damage foci, which persist over time, suggesting failure either to initiate or complete DNA repair. However, the molecular basis underlying why senescent cells fail to repair DNA damage is not known. Human premature aging Werner syndrome recapitulates many features of normal human aging across multiple tissues. WRN, the gene mutated in WS, is one of a very few genes with single nucleotide polymorphisms that consistently link with longevity in genome-wide association studies, suggesting that its importance is not limited to the rare recessive Werner syndrome, but is widely applicable across aging human populations. Despite its well-known roles in aging and DNA metabolism, remarkably little is known about WRN as a DNA protective factor in normal aging and, in particular, in cell senescence. The diagram is from a 2017 F1000 research article and depicts links between WRN and aging. This work will identify novel means to promote DNA repair in cells by restoring young levels of WRN function. Two complementary approaches will be deployed: reactivation of gene expression, and post-translational stabilization of the protein. The findings from this project will support development of anti-aging drugs acting via restoration of DNA repair in aging cells. A sensitive assay to detect methylation changes in the WRN promoter will be developed to support establishing baseline assessment of WRN promoter methylation, mRNA, and protein levels across the cell life course. Findings will be validated in human clinical samples, and a small molecule epigenetic chemical probe library will be screened for restoration of WRN expression in cells expressing low levels of WRN. Separately, phage display will be used to identify peptide motifs that bind to WRN and MDM2. The sequence of phage peptides, those similar to the MDM2 protein sequence or based on the strongest consensus sequences, showing activity in a peptide-based competitive ELISA assay, will be used as the basis for synthesis of cell-penetrating peptides capable of preventing disrupting WRN-MDM2 interactions in vitro in subsequent assays. Active drugs or peptides identified will be tested for ability to prevent onset of cell senescence following DNA damage. In cultured cells, the impact on WRN expression will be confirmed, and the agents will also be tested in cells exposed to DNA-damaging agents. Early-stage leads for novel anti-aging drug discovery will be identified based on restoration of DNA repair capacity. Outputs are anticipated to identify agents for consideration as early-stage leads for novel anti-aging drug discovery. These may be drugs, epigenetic modifiers, and/or peptide derivatives. Ongoing knowledge exchange is essential to a project with so many moving parts. Essential contributors assist with biosample provision, the epigenetic probe library, and the high-content imaging facility for screening. Early engagement has been initiated on potential downstream collaborations, for example, in regard to drug discovery experts to triage findings, target deconvolution through chemoproteomics, mRNA splicing patterns in geroprotected cells, proteomics changes of geroprotected cells, and DNA repair expertise.